Students, since we have started with the second unit from your syllabus, which is mainly dealing with the various topics related to recombinant DNA technology, I just thought to share a fundamental presentation on the recombinant DNA technology with you people, so that you get to know the basic concepts of RDNA technology, so that you understand the other topics related to this particular RDNA technology. Okay. So, we start with the RDNA technology definition. What is it? It is mainly, recombinant DNA technology is mainly joining together of DNA molecules from two different species that are inserted into host organism to produce a new genetic combinations. Okay. So, we are taking you can say joining together the cut ligated DNA sequences, and we are allowing it to join to the host molecule, and then it is allowed to replicate. Okay. Thus, the name recombinant DNA is also referred to as chimeric DNA. So, this is the basic funda of recombinant DNA technology. Next, we are going to study about a little bit of the history of your recombinant DNA technology, that it was developed by two scientists named as Boyer and Cohen in the year 1973. What is gene cloning? It can be defined as the isolation and amplification of an individual gene sequence by insertion of that individual gene sequence into a bacterium where it can be replicated. This is what you have studied in your cloning vectors. The cloning vectors were acting as a vehicle or a carrier, and they were actually resulting in the gene cloning. That is, we were isolating the sequence, the particular gene of interest from one host organism, and then we are inserting it into another one, and then the amplification was taking place. This is what is gene cloning means. Next is the basic steps of the gene cloning. What is the step number 1? A fragment of DNA containing the gene to be cloned is inserted into a circular DNA molecule, which is called a vector, to produce a chimera of the recombinant DNA molecules. See, here you can see very simple representation of the phenomena. I have given you a fragment of DNA that is red in color, and it contains the gene to be cloned, and it is inserted into a circular DNA molecule, which is, the blue is called as the vector, to produce a chimera or chimera, whatever you pronounce it, or recombinant DNA molecule. So, blue is the vector and red is the fragment of DNA. Step number 2 is the vector acts as a vehicle that transport the gene into a host cell. See, this one is the vector which is carrying the gene of interest, and it combines with the E. coli, that is the host cell, and it gives a transformed cell in which you get to have the chromosome of the host cell as well as the DNA, circular DNA of the cloned thing, that is the vector containing the gene of interest. This process is called as transformation. Step number 3 is that within the host cell, the vector multiplies, producing numerous identical copies, not only of itself but also of the gene that it carries. Okay, so here you can see that this is the bacterial DNA and these are the plasmids, and you can see the main plasmid has produced the different type of copies. That is, when a host cell divides, the copies of RDNA molecule are passed to the progeny and further vector replication takes place. Like the recombinant plasmid, transformation takes place, then amplification takes place. You can see this is one step of replication, this is another step of replication. What is step number 5? After a large number of cell division, a colony or a clone of identical host cells is produced. Each cell in the clone contains one or more copies of the RNA molecule, that is the recombinant DNA molecule. When you are producing the clone, you are replicating the exact thing. Okay, what does it contain? It contains the vectors, the gene inserted into it, and the chromosome of the host organism. And step number 6 is then the host cells are then lysed and RDNA can be separated. Okay, we'll study about the different points of restriction endonuclease. These are the bacterial enzymes that can cut or split DNA at specific sites. It is usually found naturally in a wide variety of prokaryotes, and it is a very important tool for manipulating DNA. Examples are EcoR1 and HIN3. If we go back to the history of restriction endonuclease, we can see that the first restriction enzyme was isolated in the year 1970 by Hint 2. From then over, 3000 restriction enzymes have been studied in detail, but more than 600 of these are available commercially and are routinely used for DNA modifications and manipulation in the various research work and laboratories. How do you name the particular restriction enzyme? That is the nomenclature of restriction enzyme. Each enzyme is named after the bacterium from which it was isolated using a naming system based on bacterial genus, species and strain. For suppose, look at this table. Here we are going to study about the derivation of E. cor1 name. E stands for Escherichia, that is the genus. Co stands for coli, that is the species. R stands for RY13 strain, that is the strain. And 1 in Roman, 1 is represented as first identified, order of identification in the bacterium. What is the mechanism of action? First of all, the restriction endonuclease scans the length of the DNA, then it binds to the molecule it recognizes as specific, and then it makes one cut in each of the sugar phosphate backbones of the double helix by hydrolyzing the phosphodiester bond. This is how it works. Again, I am repeating, the restriction endonuclease scans the length of the DNA, then it binds to the DNA molecule when it recognizes a specific sequence which need to be cutted down, and then it makes one cut in each of the sugar phosphate backbone of the double helix by hydrolyzing the phosphodiester bond. What is recognition site? What do you mean by recognition site? It is the site where the DNA is cut by a restriction endonuclease. Restriction endonucleases can specifically recognize DNA with a particular sequence of 4 to 8 nucleotides and cleave it. Cleavage patterns are followed. The DNA fragments are cut and they might have a sticky end or a blunt end. What do you mean by blunt end? If it is cut like this, then it is called as the blunt end, and sticky end means if you cut it like this, okay, vertically and horizontally. So this is the sticky end. Please look at the diagram very carefully to understand the difference between blunt ends and sticky ends. What are blunt ends? The restriction enzymes, when cut at the opposite base pairs, that are called as blunt ends, they leave blunt-ended DNA fragments. These blunt-ended fragments can be joined to any other DNA fragment with blunt ends. Means the blunt-ended can be joined with only the blunt ends. Okay. So here I have given you a diagrammatic representation. You can see that these are the blunt ends. You cut it like this and then we achieve this plus this. What are sticky ends? Most restriction enzymes make staggered cuts. Staggered cuts produce single-stranded sticky ends. You can see the double-stranded is cut down and we can have two single-stranded sticky ends. Okay. The base sequence recognized by a restriction enzyme is 4 to 8 base pairs in length and it is palindromic. A palindrome is a word or a sentence which reads the same from left to right and from right to left. Example, dad, madam, radar, etc. Okay. So 4 to 8 base pairs are there in length and it is palindromic, means whether you read it like this or you read it like this, it will have the same sequence. What are the sequence? A, T, G, C, U, all those are sequence. In DNA, the base sequences are read from 5' to 3' direction. If a sequence reads similar from on both the strands from 5' to 3', it is known as the palindromic sequence. So this was fundamental of recombinant DNA technology, that is how does gene cloning occurs, and then what is restriction endonuclease. Hopefully you all have understood. If you have any doubt, feel free to ask me on the Google Classroom platform. I am available there. Thank you.